I think I've generally kind of planned this out to be to go through kind of like as an introduction into bringing actigraphy or accelerometry into the behavioral sciences. So thinking about ways that you might bring it into some different health related studies or just general behavior or well-being related studies. Um, I'm not an expert on the engineering behind the accelerometry, but I can get into a little bit of what it is measuring, what it isn't, and basically how to apply this and kind of problem solve whether this will be useful for your project, or for, and also if you're just interested in interpreting data from other projects, that's kind of what this is going to be aimed at. But yeah, and that being said, since this was originally going to be in person, most of my more interactive aspects were going to be doing just that. So this is going to be a little bit more lecture style as we're walking through, but please don't hesitate to ask a question if you come up. We can dive into something as soon as the question comes up, or we'll keep going through the lecture and we can discuss everything at the end. I'm mostly going to be pacing it along those lines. And I think for some of the sections, like for device placement and how to problem solve or to do that, I might also just be standing up and trying to be on camera back here, because sometimes that's the best way to do it. But I guess with all that being said, I'll switch over to sharing my screen and my PowerPoint. Okay, so can you all see this kind of Vanderbilt branded title slide?
Yes.
Okay, sweet. So I guess, I am a postdoctoral scholar at Vanderbilt University in Amanda Leo's lab in the biological sciences. I'm an external affiliate with the University of Washington and CSDE in the Center for Studies in Demography and Ecology. Got my PhD from the University of Washington in biological anthropology, and my dissertation was focusing on interactions between stress, physical activity, and telomere length, which is a measure of molecular aging. Just to give you some like background, I guess, to how I came at this. So I guess I kind of gave this a little bit to you before, but an overview of what we'll be talking about specifically is just a quick introduction to actigraphy and accelerometry, what it is or what both these are, and when they are useful and when they aren't. I will probably switch back and forth between actigraphy and accelerometry as we go through this. If this happens at a time that's confusing, let me know, and I will be more specific and intentional about those words. But it's just in most of my work, I usually refer to it as accelerometry, but usually when you're broadly approaching it for measure the behavior, actigraphy is usually a little bit better to use. Then we're going to go into some of the implementation. So this is going to be the actual decision-making process. Like, what are some of the things that you need to go through when you incorporate actigraphy or accelerometry into your research, so that you can actually get what you want out of it. You can make it maybe worth your while, worth the effort of actually having people wear some sort of device to measure their movement. Then I'm going to cover a few of the resources at the University of Washington, mostly in CSDE in the biodemography lab. And then I'll open the floor for discussion. There are some specific decision points in here that I haven't planned a slide to go fully into detail on. Otherwise, it can very quickly become just a very boring reciting of facts. So if you have specific questions, we can jump back to things, or if we have time, I can jump back and hammer in on some other details that I think usually go unnoticed. But I wanted to kind of pinpoint some of the things that I see. I guess focus on some of the pet peeves I have in research that I had, or also some of the decisions that were more difficult for me deciding for my own research and experience in this. And then I also have some examples in here for my own research to kind of highlight where these decisions will take you. So, introduction. As a quick definition, actigraphy is generally just the use of a wearable device, so the accelerometer, to measure and analyze movement. So you could technically use, and so usually, there we go. So we usually use it in something to measure someone's physical activity. So how long maybe you're spending in exercise, how long you're spending in sedentary behavior, and then also sleep studies will often use this. And so these have become more popular over the past decade as our technology has gotten a lot better, and we've been able to have these devices to be kind of smaller and less invasive on an individual's time. They've also solved some really big problems in terms of subjective recall, because usually we find that when you ask someone how physically active they are or how much they sleep, there's multiple different ways where bias could creep in, either if they want to seem like they're more healthy, if they want to, or what they think might be the best response for the interviewer. Sometimes they just might not remember. Like a lot of people might not be getting that good of sleep or might not have restful sleep, but you might not always know that. Whereas an accelerometer, wearing that might show a lot of movement or might show a lot of wakefulness during sleep that you might not be aware of or be able to recall in a survey. And then also, I think another really part that is missed and kind of key to some of the measurements that or variables that I will often kind of propose or encourage people to use is that we miss a lot of the tiny movements that we do day to day that really add up, and there's more and more research that shows that these are also health protective. It's not just the high intensity training or the 30 minutes that you're dedicated to like just going to the gym. It's a whole range of behaviors that we can do, and a lot of times people have different recall and like how they're paying attention to it, how they're measuring it, and having an objective measure of someone's movement, like through an external accelerometer, helps us really track that. Without having to recall and kind of depend on the individual remembering it. And so accelerometers themselves have kind of gone under several stages of development over the last few decades in order to kind of measure movement better. And so now whenever you get any modern accelerometer, they'll measure movement in changes in acceleration in three-dimensional space. So actually, just to go through this again, they'll usually literally record the coordinates in both X, Y, and Z changes. And so and then it records this on the accelerometer. And then when we see it on the back end, we actually get kind of all these added up into overall movement. And so that's why we can measure a lot more about what a person is doing than maybe don't remember all the time. And so these tools are very useful for questions that are directly related to movement. So one of the major things people use this for is time use behaviors. For things like time spent sedentary versus active, you'll choose a cutoff of movement. And then people who, when they're in a period of time or when they're engaging in movement that is beyond that threshold, will be active, versus if they're below it, will be sedentary. Now there's a lot more use of total average physical activity. So this will just be the average of movement that someone engages in on a daily basis. The intensity of physical activity. So this is not should not be confused with subjective intensity. So it's not necessarily how much someone will think an activity is difficult, but it is really good at measuring maybe how much movement is occurring in a short amount of time. And then also it can be really useful for measuring sleep or how much someone is moving. Like this is also where it is really important to delve into the software side of things, depending on your specific question. And why you will also run into, I think, people using accelerometers for different things will specialize in different aspects of it. So like I'm not a sleep researcher, and so there's a lot of sleep aspects that I am not always going to be the person to ask about. But then when it comes to activity and movement and how to kind of separate these ideas in the waking time, I can help you more with that. But also I have a lot of resources and can point you in the right way of people who do really good sleep research. But so it's like, I mostly wanted to drive home here at the beginning that if for questions or variables where movement is the topic of interest, these are great. They're less useful for questions of energetics and long-term or past behavior. So a lot of people, I think, are interested in or have kind of taken movement, put that movement in a formula, and you can get some sort of estimate of caloric expenditure or energetic expenditure for movement. Um, these can be problematic for a lot of reasons. A lot of the formulas are built off of kind of an imaginary person that doesn't exist, or they need to create people, or they need to base off someone doing stuff in a laboratory that's often not going to be exactly what you're doing or what your participants are doing, and can be thrown off for all sorts of reasons. So one of the examples here that one of my colleagues is interested in is the energetic cost with walking. And even just, so here on the left, we see this kind of, and actually I'm going to turn on the laser pointer for this. Yeah, this kind of rocky path here in the woods. Even if these rocks are just millimeters high or just a couple centimeters high of just a little bit unevenness where you have to engage your foot or ankle or a little bit of maybe your calf a little bit more as you're walking. The energy expenditure of this compared to this perfectly flat sidewalk street is essentially, this could be the same as wearing like a 20 to 30 kilogram backpack. So something that be substantial, it's going to be a substantial increase in energetic expenditure to walk on something like this than on this. And an accelerometer is not going to be able to tell you that because since it just has the movement on your wrist, both of these will show up as the same. And as you move throughout your day, you're walking on a bunch of different substrates. You're engaging, you're carrying things, you're going maybe faster, like the speed, I guess it would try to pick up. But this is just how, just these tiny variations in our environment that we might not always notice could lead to huge changes in energetics. And so these, if you're interested in looking at energy and research, I'd recommend not just using accelerometers. There are other tools you can choose, like some accelerometers will have like the light, kind of like a plethysmograph, sorry, which will try to use kind of a light-based measure to see what your heart rate is, your blood saturation. A lot of wearables now have this, like Fitbits, Apple Watches, and that will usually use kind of the changes in heart rate to get a better measure of energetics. So like that can get a little bit more of how much your body is actually working. So say that still won't always do great in terms of catching the differences between between these two substrates, but we can talk more about that if energy is one of the things that you're interested in with movement. We can talk about strategies to make it more or less invasive and for people and helpful for your research. The other question that, or the other issue I think with this, or something that they're not good at answering, is kind of your general behavior or your behavior in the past. We're only able to measure the behavior for a limited amount of time, and this might really sound like a dumb moment. And many of us, like if you've engaged in this, or if you've started wearing a smartwatch that records your behavior or like your movement, then you might know about like just generally trying to increase your physical activity so that like you look more active. So we all kind of get that, and like if you think that you're wearing it for just a week, it's like of course someone might change their behavior for just that week. But a lot of times we're interested in questions where there is kind of a long-term feedback between physical activity and say biology. And so I do think that this is something that you really need to keep in mind when looking at this type of research. Like it's something that I've had to contend with when I've been really interested in looking at if physical activity can moderate the impacts of, say, psychosocial stress on your biology or health. And I only have a week. I kind of have to hope sometimes that week is a good proxy for that person's behavior more broadly. And if you have a lot more people, maybe you'll get better at that, but this is always something to keep in mind. And then maybe try to use other parts of your experiment or research design to make up for that potential caveat. Okay, so all those kind of bigger picture things being said, let's jump into some implementation. And here there will be some parts that I skip over. So if you want me to talk about them, we can pause and we can talk about them in more detail. So when you're thinking about how to implement a study with actigraphy, there's going to be three main things that are kind of be feeding back on each other. So the whole design process isn't really like a solve this, solve this, solve this in like a really ordered fashion. You're probably going to like just make one decision, then that will help you make the next one, which then you might have to go back and forth on. But so I'd like to think about it as being split into three main categories. You have some questions related to data collection that you need to solve for yourself. You have some decisions that you need to make regarding data processing, because there is a lot of raw data you get from these watches, and there's multiple different options you have for actually processing the data that can impact what you actually have as the end result. And then there are also some important decisions with the data analysis itself. So once the data is processed, how do you actually operationalize it for your analyses and your questions? And then, like I said, I just even right now went through as like a 1, 2, 3, but all these will keep going back and feeding in on each other, which I think will become clear as we move forward. But the first, just because I need some sort of order to walk through these, we'll start with data collection protocol. And some of the major questions here that I think people always need to consider is the participant burden. As with all aspects of a research project, actually I'll get onto it next. I'll just be covering aspects of participant burden, device selection, device placement, sampling frequency. Oh, yeah, and that's that. And then, yeah. So first, participant burden. Here's a picture of myself wearing an Actigraph. And as you can kind of see, this is one of the, I think, higher research grade accelerometers you can get, but it is big and bulky. And so when you're going to ask someone to wear this for a week, you should consider some basic things like their comfort. Is this something that someone is going to be able to wear for a whole week or however long you want them to wear it? How are they going to be worried about caring for it? So sometimes I think I can tell you from experience that not all participants care about the object that they're wearing, and some of them might misplace it or not even care to return it to you, which I guess is another aspect, is make sure that before you start giving these to people, that you have a good plan for receiving them back. If you're shipping them in the mail to someone to work remotely, make sure there's some other way of ensuring that it comes back. And then also aesthetics. And this kind of cuts both ways, where someone might have to wear this Actigraph. If you want them to wear for a full week or reliably, you have to think about their positionality, what their plans are for that week or for that time. Is it something they're probably going to wear? So like if you're, let's see, like I think if we're going into the weekend and you're having college students wear this, is this something that people are more likely just to take off when they go to a party? Is it something that then will become like a conversation piece? I guess it can work both ways there. And then also, if it looks something like this, where it's just a big old red box, then also people are less likely to steal them. So it can work both ways, and both you want them to have good, you want participants to try to wear it, but then you also want things to come back. And so some of these decisions might also be answered, like if you have enough funding to say like give people Fitbits after wearing it for the week, then maybe you might need to worry less about aesthetics, because it might be something that already fits in. And if it's like, I think some of the consumer grade things, in general, the participant burden is going to be a lot less, because it's designed to be something that people will want. But I think for a lot of the research grade stuff, really consider whether the burden, one, is worth it for the participant and the knowledge that you'll get out of it, and then two, also the practical thing of them actually wearing it, because you might not get good data or any usable data if the burden is going to be too high for them. With that, I'll jump into device selection. And so I do want to start out by saying most modern accelerometers can be used to produce comparable results within a study. So some of it will depend on like how you operationalize the data you're going to use. But for some downstream processing of the data, as long as it's a device that you can get the raw accelerometry data from, there's a good chance that you'll be able to at least get something that's similar to another study. You might not be able to directly compare these all the time, like exactly say exact intensity someone is been doing in a very specific I think like activity. But if you're looking within your group and within your sample, you should find similar results regardless of what accelerometer you use. If all other aspects of the protocol are the same, right? Like, so this isn't to say like a Fitbit on your wrist will get the same thing as an Actigraph on your hip. Those two placements will give you completely different results, and I'm going to do placement actually in a slide after this. And so usually when considering what kind of device to use, some example questions, these are not all the end, like I think these will kind of shift depending on your goals. But you want to consider how many devices do you need? So basically and so how many devices do you need versus how many can you afford? And like what's the way to optimize that? What devices are available to you already? Say if there's a shared institutional resource that has these that you can check out for your research project, you don't need to spend money on them. But as you can see, as are spread out on the table in front of Tiffany in Rate Hall, then that can make things a lot cheaper and can also help give you nice, reliable results more easily. And then I guess what is the most affordable option? I think this is, so in general, price for these can be one of the biggest hurdles. I can speak from experience on this to getting accelerometer data, and which is why you'll see some of the labs that do kind of keep pumping out like actigraphy results or accelerometry data, often will use the same devices for a long period of time, and you'll use them until they pretty much die. They start slowly dying, or you start losing them. You'll just take them as long as you can go. And I think for the most part, given our modern processing abilities with some open source software, that's fine in terms of what you're going to be getting for your research. So now placement. I think this is one of the things that my first answer to this is there's probably no right answer. This will depend on your research question. Some of the three major options for this are having someone wear it on their hip or on their waist. Usually with this, you'll have them either attach it directly to, say, their pants, or in the biodemography lab, there's some larger waist straps that you can attach, that people can attach it to. It'll be on their hip. This is pretty good if you want to strictly see how much someone's entire body is moving. So like if locomotion is really important, if how ambulatory someone is in their physical environment, this will be pretty good for that. I think it's less, but one of the kind of barriers you can see here is that it's going to be harder for people to wear this, maybe perhaps, or it could be harder for someone to wear it all the time. So for example, I study athletes, and there's a lot of maybe movements or sports where having it sit on the hip like that might be dangerous or get in the way, and even maybe more so than something on the wrist. So they'd have to take it off for that. Or also, it's really uncomfortable to sleep with these. And if you want the results to become comparable between individuals, the placement on the hip is going to be really important. It's going to be really important that someone keeps in that general area. So since it's measuring how it moves in 3D space, when you're processing the data, usually you tell your program where these devices were placed. And so having it placed on the like outside of the hip versus in the small of your back, like if it rotates, will give different results. And like the kind of measured acceleration or changes will differ based off that. Then I think the most popular one is setting it on the wrist. And I think we can all think very quickly of ways that this could become biased. I'll start with the cons and I'll go to the pros for wrist, because this is the one that I usually lean towards. Is that I think if you've ever had a smartwatch or something that you have a movement goal or whatever on your wrist, and you really like are kind of near the end of the day and you want to reach that goal, or if it's like yelling at you to move, you don't really want to, you can just like move your, like flail your arm a lot, and it will start recording that as movement, because that's where it has. Like I think there are like the algorithms that are in there will try to correct for general movements of the arm, and will give you different results if it's on the arm versus the hip. Like if you moved So through some of the filtering processes, if you tell them the actigraph is on the wrist versus on the hip, or if you say that it's on the hip and then like it sees movement that's like incredibly like fast over the head, it's gonna not really know what you're doing, but it's gonna really impact, like that's gonna bias the movement up really high, and that person's gonna look incredibly active and kind of berserk in that stance. But and then also we'll bias it towards um, something just in general. You can move your hands a lot without moving your whole body. And so this would be another one where caloric expenditure would be a difficult measure because your hands are going to be doing a lot of things throughout the day that your body isn't. However, most studies now will typically go with wearing it on the wrist and on your non-dominant hand. And so this will usually reduce the chance of error between, or kind of reduce the kind of movement for someone who's just sitting still, since it will more likely use your dominant hand to do things that you're around. And then also, if everyone has it on their non-dominant hand, it's usually going to come down to it's just going to be added noise. So you can still see the differences between people, unless you think like the only time that it becomes an issue is really if you think within your sample, people will be using their hands or arms differently. And I have an example of this with athletes that I'll come to later. But in general, like wrist allows someone to keep it on for the majority of their day. It is not particularly pretty to look at, depending on the device, but it's readily accessible, and someone can like, I think it seems to go with pretty good results being on people's wrists. And then another one, which is I think much less popular than either of these two, first two, is wearing it on the ankle. There are also ankle straps in the biodemography lab if you want to do this. But for me, I've usually mostly just used those for people whose wrists were too big for the straps, for the wrist straps. But because this one, I think probably would have the highest rate of error if you want to have some, in terms of like it getting hit, getting it knocked off without someone noticing, if you have someone wearing it for a long period of time. But similar to the hip, your feet probably aren't moving a whole lot more than you are. So you are probably going to get maybe a tighter relationship to when the whole body is moving compared to the wrist. So one of the more boring things that I think you have to select when you're going through this, but one of the things I think is always good to highlight, because it's one of the things that isn't always even reported in the literature, is the sampling frequency. And this is something you need to set before you give someone their actigraph. And basically it's setting up how often per second the position and acceleration is recorded. They generally, I can use the actigraph WGT3X as an example. It will go between 30 hertz and up to 100 hertz. And this means like if it's collecting data 300 times a second up to 100 times a second. And for this, I think most people nowadays will recommend trying to go up as high as you can from what will work within kind of your data analysis software, because it just gives you more data. As long as you have everyone use the same measurement, you shouldn't have too many biases across. But it's just that higher acceleration should give you a better chance of recording more data if someone is very highly active. So like if you're studying athletes, having a higher hertz might give you more accurate differences between athlete groups. Or if it's a lower hertz, you might actually miss some of their higher intensity movement, because it might just clock itself like at different times. It might look like less movement than it actually is, as if it was like collecting at 3 times the speed. And that kind of has here, you also get more analytical choices with the higher hertz when you go down. That can make it more sensitive. So I think as long as if you're not worried about battery life, try to choose the highest hertz that you can get, or highest sampling frequency that you can for your device. And so with that, is there any, like, so these are all decisions that are made kind of before you give someone the device, and now I'm about to move on to things like that you get after the decisions that are made for when you get the device back. But are there any of these questions here that someone wants to dig more into, or I guess challenges or things that just don't that also don't sound right? With that, I'm happy to go into detail before moving on. Okay, I'll accept that as a not yet, Rob. I'll ask later for those. So we'll move on to data processing. And so these are decisions that you can make after someone has worn your device. And so you've gotten it, you've plugged it into your computer, and you're going to start downloading it into, if it's an Actigraph, you'll be downloading it into Actilife software, and then working from there. So some of these decisions, I won't be getting into all of these, but I can go into them at the end, because there's a lot of details and decisions to make. You'll have to make decisions on non-wear time. So basically this is some thresholds you'll set in the software to decide when, and algorithms that you can use that will decide when someone's not wearing their accelerometer. And then so you can know not to include that. And if someone isn't wearing their accelerometer, it might just look like they're sleeping very still. And you want, and you want to make sure that you don't use that to kind of deflate the amount of activity someone is engaging in. You want to define different terms for what incorporates a valid day versus valid week. So this usually has like how many hours per day, how many hours for a specific day does an individual need to be wearing their device for you to count that data. And so this ranges, I think, this will change based off whether you're having someone wear it all day, like when they're awake and when they're asleep, versus if they're just asleep or if they're just awake. But like if you're having someone wear it for 24 hours a day, now I think the general consensus is to look for days that are like 16 plus hours. And then, and so that is if someone was wearing it for only 15 hours that day, just don't include it. Otherwise you might get biased. You might bias your data compared to some of the other individuals that are in your study. There's also filter settings. This is pretty much, so a lot of that happens under the hood of whatever software you're using. And you'll often, even if you get a choice, it'll just be two options for that. And that might depend on exactly what you're looking like, the group that you're working with. I think I'll speak for Actigraph on this, that they have a low frequency filter. I believe that's the type name now. I think the specific name is escaping me right now because I don't have it written down, which was a mistake on my part. But the regular filter is usually going to be good for whatever you want to do. I think there's some settings that will sometimes help you save battery life. So if you have people wearing these, or you want people to be able to wear something for like a month straight, you might do that so that it can switch into energy saving mode at times. Epoch length is something that I don't have on here, but we can go more into why this is really important. This is basically when you have the hertz is going to be like collecting data however many times a second, but then epoch length is going to be saying what's the like unit of what's the base unit of measurement that you'll be looking at for your study. So usually a common epoch length I think is like 10 seconds. And so what that means is that it will average the acceleration that happened every 10-second block. And then that block will be given a score if it's the Actigraph software likes to use counts of activity, which is their own proprietary measurement. So they'll give each 10-second block a certain number of counts of activity, which was kind of the average number of counts that it passed during that 10 seconds. And then sleep-related behavior, I will be highlighting that in a second. And then available software and programs, I'll go more into detail on that on its own slide as well. So for data processing, I like to highlight that sleep is actually a really important decision, like in how you're measuring and defining sleep is a really important decision for you, even if you're not interested in it. Or like you're not interested in it as it being like your main outcome measure. Like for me, I was really interested in how active someone was throughout the week, like maybe how like the intensity or average total physical activity they're engaging in. I wasn't super interested in how well everyone was sleeping, even though I know it's important for biology. It was mostly just I was interested in it as a control variable, but it is also central just to even defining your physical activity. Um, and like first and the very basic thing is that you needed to separate um, awake, you need to separate sleep from awake and sedentary time. So, because there will be times when you want to know if someone is engaging in sedentary behavior, so like they're awake, but perhaps just not that active, versus when someone is sleeping, because either one might give you a better denominator upon which to like see how much time, either what proportion of the day someone is engaging in that activity. Say like if you're interested in how much of the day someone was spent in moderate to vigorous physical activity, you want to have the correct denominator of how long they were awake during the day um, in order to get that proportion. Um, so like even just in this, when you set it up, like right away, we'll also use that to kind of filter in, like all these software programs will also use that to like um, on more of the heavy math side, which I will not be able to speak too much on. It'll also use that to help these estimates from that point. There are multiple ways of defining sleep. Um, so I think there's, so I think my best recommendation for this, if sleep is not your variable of interest, but you're looking at it like as, but you want to make sure you're controlling for it properly, or in a kind of powerful way. There are some consensus statements out there where people are like highlighting which one they want to do. Basically, these algorithms just take a certain amount of you portion out how much you're willing to, how much like light, like very very low activity you're going to accept and define as sleep. Um, People who are studying sleep and like the most, I think the most powerful way to actually look at this is to have both a sleep questionnaire and have the like an accelerometer of some sort. So like those, like either the next day you'll say what time you got into bed and then what time you got out of bed, and then you can use that to validate when your device was saying that person was asleep or not. Yeah, and then there are multiple measures of sleep. So uh, I think as someone who is highlighting and specifying and like specializing in sleep, there's going to be a lot more things for them. Um, but using these algorithms does give you a pretty good measure of, say, like total sleep and wake after sleep onset that are well validated against things like polysomnography or other lab-based measurements of sleep. So you can usually rely on it for like the total hours of sleep someone is having. So then like what the average is sleep, and then also um, wake after sleep onset is basically how many times do you wake up in the middle of the night after you fell asleep. Um, these are both like measurements that are pretty good based off this. And then there's a kind of sleep efficiency, which takes, I believe, both of these into account, as well as the amount of time then that it's estimated that you're awake after like the major going to sleep time and major waking up time. That are all pretty powerful covariates if you're interested in someone's like, if you're interested in looking at activity or other movement based things, you want to use sleep as a covariate. So with that, I'm also going to jump into what I think one of the biggest decision points that you can make when you're going through this and working with your data from accelerometers. So first, and this is like choosing which software program that you want to use to create the variables that you're interested in. So most accelerometers that you get for your research will have some sort of proprietary software that is associated with them. You often might need to purchase this license on top of your devices. I think the Biodemography Lab still has the license for Actilife, which is Actigraph's data analysis software. And so there's pros and cons to just sticking with the Actilife software. The pros is that it's easy to use. They try to make these user friendly. They want more people to use the software to look at the research. And so there's a lot of ways that they make kind of quick comparisons you can make between groups. You can do a lot of your analyses just within that software. I mean, you can't maybe do like as fancy as statistical modeling as you might want, but you can do group comparisons. You can find means. You can do a lot with it very easily and quickly. And then also, what I think is one of the great like kind of ways as compensating your participants with accelerometers is to share the data back with them. Especially some participants are really, especially if someone has, say, a smartwatch that's recording their behavior, having this kind of research grade accelerometer is really interesting to a lot of people to have that compared to what they're seeing. I will give like one caveat is that I think a lot of the consumer electronics that we wear and we use to analyze our behavior will often take shortcuts and make assumptions and tell you data and information that probably isn't right. So sometimes they might be, so some people might expect more data from these than they're going to get. But either way, it's, I think, like doing things like estimating moderate to vigorous physical activity, estimating steps. These things are really interesting to see how they can sometimes differ from someone's consumer grade wearable. And these proprietary softwares make it very easy for you to print these out. You can have someone come in, have them fill out a survey, or even just hang out in the waiting room in your office for just 5 or 10 minutes while you just upload the data. And then if you could save templates that you can just essentially press print, it will print you out a PDF of something that you can just essentially either email or hand to or walk through with your participant right there. But then this being said, like this, for all this easy use, some of the cons are that a lot of these will rely on proprietary measures that they don't really tell you what goes into calculating them. So Actigraphy in particular will rely on counts of activity, which is something that is kind of abstracted from the raw accelerometry data. And some of these, like in order to make it more, I think, digestible, they do this kind of chop off your movement. So like you do, some of these may disregard or underestimate or overestimate movement because it's constantly looking for a cutoff, like either above or below where you are. And people outside of the company you're not really able to double check how they're calculating it. It also only works with the same brand. So if you have Actigraphy, you can only use Actigraphy with Actigraph devices. So if someone has an ActiWatch, or if someone has like an Activive or like or Activity or any of these other brands, you can't use them with this software. It all has to play within the same company. And so I guess what are your options outside of that? The major option that I think and more of the research world is also turning towards now is this open source R package called GGIR. And so the pros of this is that it will work with the raw acceleration data. So you could go and double check all the math that is actually happening in this package. It also integrates well across accelerometers. So earlier when I was talking about how a lot of modern accelerometers should give you the same results. If like, say, if you deployed an ActiWatch in everyone in a study at the same time as an Actigraph, as long as you use the same protocol for both, you should have the same or at least very, very comparable results. That's if you put it into GGIR, because it will take the raw accelerometry data and you can control the process from beginning to end. And you don't need to understand everything that's happening. There's a lot of automatic options that do come here, so you don't need to become a full GGIR nerd to use it. You do need to know, it does help to know how to use R though. And I think there's just a lot to be said that this is also a better way to make sure that a lot of our data compares across studies. Whereas like if you have Actilife in one study and then you have another brand in another study, in a different study, it's going to be it's unclear how well we can compare even the broad results across those two software companies. But some of the cons are there is a steeper learning curve. So although you don't need to become an expert in it, it does require, or at least every time that I've used it, it does require some R programming skills and does require someone at least to sit down and devote some time into selecting the options that you want. And then also it's not immediately available to provide data to participants. So it's not going to be as quick as Actilife's just kind of like save print. So to kind of use the best of both worlds, what I've done to kind of solve both of these is that I like to use Actilife to be able to share data with participants right away. But then when it comes to the actual processing and analyzing data, I like to use GGIR to give me my data on that end. Okay, I'm going through this much slower than planned. So is there any questions on this data processing part? I realize that this is also might be more the most boring part for most people when thinking about accelerometry. Okay. I'll try to go through data analysis a little bit more quickly so we can just get to people if they have specific questions about their own research, we can try to answer that stuff. Data analysis, excuse 1 second, I'm gonna take a drink of water. Basically going to cover what the main outcomes or variables are that people usually use and that you can decide to use. And then you often need to make some really big decisions on the data analysis side for modeling and hypothesis testing. I decided not to get too far into that for today because I think that is probably going to be a thing that's more out of, all this is very specific. That's probably the one where you can go many different like even more different directions than you can after this stuff. And some of this is so far things that will also help you kind of compare and understand like the decisions someone has made in the literature. But so data analysis, this is one of the most raw outputs you can get without the numbers from GGIR from an accelerometer. And so I'm using this to more highlight all the different ways that you can look at this data. And I'll refer to it at least in the next slide. So here you can see that it has both a measurement of the angle of the z-axis to a horizontal plane, and it's using gravity to kind of sort what angle it is and what is horizontal. It also has arm movement, which is just the change in like location. And then you can see kind of just the graphs of the movement of these two variables changing throughout, as well as some just immediate categorization of different activities. So like, and these are thresholds that you would set in GGIR before it starts. So like looking up here at our kind of what these color codes mean. Down here is like this x-axis is time. And then you can see basically that this gray is when people are sleeping. So this person woke up at a little bit before 6 a.m. and then went to bed a little bit after 8 p.m. This was, and then actually accidentally copied the same one twice. So I guess ignore the bottom one because it's just a repeat. And then you can see that like how each essentially minute spent awake in like the major waking time period is separated into this purple, which is inactivity. So that might be what someone would call sedentary behavior, like maybe they're sitting or maybe being like very lightly moving. And then there's periods of moderate physical activity, or light, which is just really light, and then moderate. And then vigorous, which it's hard to tell in the color here if we actually see that in this person's day. But you can see that each minute is accounted for. And even though like there can be long blocks of time with the same color, there's still a lot of movement within that same color. So like this like let's say some of these lines here, whereas this is arm movement, there's actually some periods where that arm movement goes up above what has just been classified as moderate physical activity here. And that's partly because sometimes for these blocks, you want to set a minimum amount of time in that block, and that kind of helps get rid of some noise that you might see from like maybe someone waving their arms or gesturing if they're giving an online like workshop and they're moving or gesturing while they talk when they're really just sitting. But so this is a lot of data to look at and to try to understand. And so the next two slides are just going through some popular ways to make this understandable and something that you can actually throw into your analysis. And so the first thing, I really thought I had two different things set up, but I did not. So for the categorical measures, so these you're gonna, like most people will have heard of this. This is gonna be like the time spent in moderate to vigorous physical activity. This is something like time spent sedentary. It's something that I guess I'll go right here. It's easy to conceptualize. It's easy for us to think about like in categories, like if someone's walking slowly or like you're walking kind of at normal pace versus if you're jogging versus if you're like sprinting. It's easy to communicate this. So it's easy from a public health standpoint in particular to be like 30 minutes a day, this will be associated with this many health outcomes or like these positive health outcomes. But some of the cons are that it can miss important movement and variation. So in this grouping, like the algorithms aren't perfect. So like to even highlight some of these, like there might be mischaracterizations here. Like someone might be doing something here that is actually classified as inactivity or it's in this inactivity block, but they may have actually been active, but just for a very short amount of time. Like if someone got up and like, maybe if you had to rush to catch the bus and like had to sprint a couple of blocks, that might not actually take up enough time to actually change into a different threshold. So that whole time period that you were in there might be inactive time, by the way that most of these algorithms work. They're also perfectly collinear with each other. So if you're doing a data analysis, you need to make sure that you don't include physical activity with sedentary time all the time and with sleep, since they're all going to add up to one. So if you increase one, all the others have to reduce in some way. And this is the part that I think usually if we're trying to control for say like the impact of physical activity, you might try to throw in moderate and vigorous as well as say light and sedentary. But they might be like messing up your models because since they all vary, they necessarily vary with each other. And then also one of the more important parts here is that these are arbitrary cutoffs. To go back to that movement is bad at estimating energetic expenditure. A lot of these cutoffs were created based off of the calories someone was burning, I think moving at like a certain rate. Who that person was, we don't always know. Was it a marathon runner? Was it an Olympic sprinter? Was it someone who like me is sitting at their computer most of the day? All these things are going to change how their energy, how it's kind of expended. And also even within that, based off of like whether or not it would be predictive for you or for your participant, the cutoff itself was more or less arbitrarily chosen. So those things like the fat burning zone versus carb burning zone, which we'll sometimes run across in like more popular exercise things, are all more or less kind of arbitrary. Like there's not like a specific, like a certain like movement once you get to a certain point doesn't all of a sudden automatically become a different type of metabolic pathway that's being incorporated. Like there might be some broad changes, but like it's always going to be more on a continuous scale. So when you're thinking about the biology, you might miss important things by using cutoffs because the biology, these cutoffs were not built off of the biology. They're mostly built off of our convenience as researchers. They can still be very useful. Like it's incredibly useful to be able to communicate the benefits of exercise and having 30 minutes of activity a day. But you need to make sure that you're either caveating that message or that just when you're designing your study, that's going to be a potential thing that weakens the association you're looking for. And this, I'll say things that I'll bring up some of the outcome variables that I'm overly excited for, and was one of the things that has kind of kept me in this research with these coming up. And I don't expect everyone else to be as excited, but I do want to say that you should consider these like alongside the typical categorical cutoffs. And there's a lot more. I'm just going to share the two kind of easiest to conceptualize ones. I can go into more of these if, depending on people's questions. The first is average acceleration. And so both of these are measures that are coming out of that GGIR world. So like once we made the open source platform where people could actually deal with the raw acceleration data, then there was kind of a plethora of these different outcomes have come up. Average acceleration is one of the ones that has been the most published out of this group of measures. And basically it gives us a single measure of total activity. So it can be considered, I think its closest relative would be steps, like a step count. Except obviously if it's on your wrist, it's going to be giving you more of your movement than actual, like maybe full steps throughout the day. But that can be both helpful and hurtful. So like if you're a rower, if you're on a rowing machine, if you're on something else where your arms are actually doing quite a bit of work, this will be a better job of catching that. I also really like this because it's all of the activity. And I think in more research that's coming out with this, the UK Biobank has been publishing there have been a few studies coming out from UK Biobank using this that are really like reinforcing the idea that any increase in activity can be beneficial. Of course, this is for most of us, there is definitely a point where you can be too active and it starts hurting you. But in general, for most sedentary populations, this has been a good marker for just showing that. Like you don't necessarily need to do that moderate to vigorous physical activity to have health benefits. And if you use those categorical measures, you might not catch those benefits. Whereas this is just like any movement that you're awake, the average of that is associated with some good different health and biology. The other one that I really like to use, and I think is often used with average acceleration, like since this total activity doesn't tell you like how maybe intense that activity was. So like for average acceleration, if someone has a high score, you don't know if that was built up from like, was it 10,000 steps in a day? And like that you just kind of were just regularly moving throughout the day, or was it because you had a really focused hour of activity? And so both of these may impact someone's biology differently because they may incorporate different like metabolic pathways. Like if you're engaging in intense activity, it's more likely to engage like an inflammatory response, cortisol response, like testosterone responses that might be impacting your biology different than just overall activity. So a good way then to kind of combine the benefits of that moderate to vigorous physical activity is to use both of these metrics. And without going into too much detail for the intensity gradient, is that it is also a continuous measure of time spent at different intensities. So it is literally the slope of the lines. I'm going to get my window out of the way. It creates the log of both the time accumulated. It takes the log of time at a different intensity and the log of that intensity, so that you can get, because usually if you didn't log it, you'd get a distribution that was probably like this, where it was like a curved line where people will spend very little time at high intensities and a lot of time usually at low intensities. So when someone's slope is more negative, like say this, you're spending less time at higher intensities and more time at lower intensities. Whereas if your slope is flatter or less negative, or I guess it could also be more positive, but if you have a flatter slope, then that means that you're spending more time up at the higher end of intensity and less time at the lower end of intensity. Um, yeah, and then to go into like how these three different measures like compare to each other, I actually have an example from some of my own data to share. So this was data from college athletes playing in a variety of different sports. So first, I'll show the time spent in moderate to vigorous physical activity. And so here we have non-athletes in this first box plot. The y-axis is the actual time spent in minutes per day. So if someone said it was at 100 minutes for moderate to vigorous physical activity, that was their average time spent per day in moderate to vigorous physical activity over the course of the week. So you can see that non-athletes are right here. One of the lower groups, I think, as you'd expect, because they're not Division I athletes who need to train for their job. Track and field athletes are kind of the second highest group. Swimming is the same as non-athletes, and as you could probably guess, or maybe not, but the Actigraphs we were wearing, we asked them not to bathe or shower or swim with them on. So this is another thing to consider when thinking about physical activity, is that are you going to be able to measure the activity that you're actually interested in? So these swimmers, I have no idea how active they were in the pool. They could have been swimming for hours on end, but without getting good questionnaire data, I won't be able to like really connect them in some of the other ways. Unless they've reported that they weren't really exercising that week. So say these weeks were like rest weeks, I could still try to see like how that is associated with inflammation or something. But then we'll jump up here to the group that was clearly the highest. Rowers spent by far the most time in moderate to vigorous physical activity. This person right here was a coxswain. So whenever they're on the boat, they're not rowing. So it makes sense that they'd be kind of in the distribution that everyone else is in. And then other here was mostly track athletes and a couple tennis athletes. Got a head track over there. These were like, this track and field group is the runners. This had some throwers and had some event track and field athletes, as well as tennis, and I think and a handful of basketball players, but it was more of a grab bag of kind of the smaller ends. But then when we look at average acceleration, so just an average of all the raw accelerometry data that someone collected, track and field then becomes incredibly diverse, but has the median and I guess, sorry for not describing this earlier, this bar in the middle is going to be the median of this, and then the 25% quartiles on that for the box and whiskers. So as you can see here, the people that were actually track and field is by far higher on here. And the y-axis here is not time in minutes, it's milligravitational units on average. So once again, this gets to why MVPA is much more easy to communicate to people, because I actually do not know if I could tell you what 100 milligravitational units on average feels like. But as you can see, like depending on which one of these two just measures of general physical activity you use, you get two very different stories when you're comparing different sports or just people doing different behaviors. Because here rowing comes down here, there is still a significant difference between them and these other groups, but it's not nearly the same, and track just becomes much higher than them on that. And then we see a similar thing when we look at intensity gradient. So remember, this was the continuous measurement of intensity that you engaged in. So while and these were two, and actually I think these two were the ones pulling this down here. They were injured for part of the week. So they weren't training. The intensity gradient is that slope. So people that are higher up here have a flatter slope. People who are lower have a steeper slope, which means that they're spending more time at lower intensity levels. And here, similar to average acceleration, track and field runners are much higher. So they're spending more time and the continuous measure of intensity at higher intensity levels than individuals rowing, or than it looks like they are when they're in MVPA. So whereas rowers here are much more comparable to some of the other groups when you look at the intensity gradient. And I really like to think about these graphs when I think about like the data that people present, because these tell you very different stories. If you're looking at just moderate to vigorous physical activity and you're interested in athletes, there's a clear difference where rowers are much higher. But if you take a combination of average acceleration and the intensity gradient, it flip-flops and track and field is much higher. I have some theories for why this might be the case, but and I think part of it is actually how all these are kind of measured, where it's like because track and field runners are running and have a much less maybe consistent tempo than you would as a rower in a boat. A lot more of their most intense activity would have been faster and at levels where the accelerometer was actually accelerating and moving much more quickly than a rower would ever move their hands while they're in the boat. Since the goal of a rower is to move the boat as fast as you can, and you need to sync your movement, whereas when you're running, you need to keep moving your arms as much as possible. So that's another way where device placement, the behavior of participants is really important to consider when you're interpreting your data and also when you're choosing your output. So like for me, I like to use, because I have this result, it's probably better for me to include all three of these, but even include this categorical variable since it might be telling us something that's really important about their behavior. But when you are reading through a study or you see a headline that's talking about just time and MVPA, I just ask that you remember these kind of side-by-side graphs where the groups literally shift in comparison to each other based off that measurement. Oh, and then here was just kind of more direct comparisons with just the rowers of track and field, just so you can see a more drastic shift between the different measurements. So yeah, so as you can see here, like a lot of these will go back and forth across each other. Like, so your data analysis, if you know which measurement, what you want to use, then that might inform where you want, say, like what device you want to use, where you want to place the device on someone. But then also data process, but it will also impact the data processing. But then your data processing, if you're limited there, will necessarily determine what data analyses are available to you. But so it's when you're going into your project or thinking about what you want to do or interpreting someone else's project, thinking about the relationship between these three general categories is important. Just a quick slide on the resources available at UW and the Biodemography Laboratory. I think I've already brought most of these up, but just as a reminder, there's 10 plus Actigraph devices there. We used to have more, but I ran on a remote study and was not very good at making sure these came back. I have other better strategies now for everyone else to use so that we can make sure we don't lose any of our shared equipment in the future. And then there's personnel resources. There's a great director of the Biodemography Lab that we're all very lucky to have access to, and Dr. Tiffany Pan. And then if you ever want to go through some of the growing pains of any of working with GGIR or anything else with me, or feel free to contact me. And even if you just need emotional support, because some of the people working with GGIR are kind of much more deeper in the programming than say I am, and I think many of the people using it are not nearly that level, but so it can be difficult sometimes getting help and stuff from them, which is again one of the cons of that steeper learning curve with them. But anyway, feel free to reach out to me anytime after this. Consider me a resource, especially because I realize I talked much longer than thought. When I write these slides, I always underestimate how much I go off script. But I did want to leave some time for discussion or questions anyone has on this, or concerns, and even if you have a specific question for your own study or idea, feel free to ask.
Thanks, Rob.
Thank you, Rob.
Thank you, Ken. Okay, yeah, thanks, Rob. That was really great. Are there any questions? I don't see anything in the chat, but if you are on Zoom, feel free to unmute yourself and ask questions.
Yeah, I have a question. Hi, Rob. I'm Enza, a doctoral student at University of Chicago. So our project team starting to use Actigraph for measuring sleep. And I actually had a personal experience using it and then get a clinical report from the
Okay.
ActiLife software. But the hard thing that I encountered is that the clinical report was not that self-explanatory. So I was wondering how you share the sleep study or the data with the participants.
So I usually first I'd want to like I walked through like after I had it pop up in the lab, and so I'd walk through like say I'll jump back to it's not the same output, and I meant to put that in this so I could walk through something like that, but I'll just use this as an example. Like and like so you had that thing right that was probably may have had some summary data like your averages over the however your time period was, as well as like a weird like thing where you see like to give everyone else some context, they sometimes it runs on like this, like, and you're never really sure. Like, it can be confusing, like where the day begins, when the next day starts, like what was actually asleep. Um, I think like for participants, I did walk them through it once, and like tried to highlight like this is so kind of like what I did here, where I was like, this is like this gray areas where we found that we were asleep. Um, Here were some times when you're marked as like not wearing it, or there were some times when like also I think sorry, just to go on a side tangent with this too, is to walk through them first. Like one, it will help them be able to read it because like you said, it can it's not always super self-explanatory. Like you can have it print off some summary statistics for them, but that's not always super interesting for people. But as you walk through it with them, it can also see if something was coded completely wrong. Like, so I had someone who um, got like coded as not wearing their Actigraph for like a good 6 hour stretch during one day, and we were able to catch it because um, I was walking through with them, and it was like, oh, okay, and then looks like you took it off here for a break, and your Actigraph wearing, and then it did help us find an Actigraph that was defective. So, but I think but in order to just go back to all this is I think I recommend walking through it if possible with the participant. I also if you could like I also make like a PDF like explainer for people that basically was just a very like thing like the, cueing them where to look on the thing. So it kind of sucks to make it because it takes a lot of time to kind of explain and write down how someone should read these. But like once you do it once, then it's easier going on. But I'm sorry if I don't know if I fully answered your question here because I kind of been distracting myself by clicking back. So what were some things I could touch on a little bit more for you on that?
Okay. Sounds great. And thanks for introducing GZIR. Even didn't know that it's existing. Yeah, thanks. Thanks for your
Oh,
presentation.
yeah, you're welcome. Yeah, totally. It's great. And I'm also welcome. I can also try to share I'll double check to see if I still like any of my explainers for my participants. Otherwise, um, yeah, you could probably, especially if you're focusing on sleep, you might have specific things that you want to highlight for them specifically on that I probably won't know as well for the Actlife software as you do.
Sounds good. Thank you.
Okay, it looks like Carly dropped a question in the chat. Do you have recommended resources for considerations? So like, I'm guessing, so is this like trying to decide between them or like trying to see like how much the costs are or
Sorry, I don't know how good my audio quality is because I had to join from my phone. Can you hear me?
Oh, gotcha. Yes, I can.
I have access to Garmin and or Fitbit data from secondary data sources. So I guess I'm curious more about analysis and the limitations to be aware of in analyzing existing data from those sources.
Oh,
That's still a very big question. So
yeah,
whatever you can share.
off the top of my head, I don't have like, I think great resources other than maybe GGIR might have some good recommendations if you want to use um, them for like, yeah, working with the raw acceleration data. Also in general, for everyone, I guess there is, and now I can't remember their first name. There is an author that does a lot of consensus work, does a lot of kind of descriptions of these newer techniques and newer models. Last name is actually, I have his paper pulled up in another sheet, so I'll pull up his full name to that like if you ever want to like just find some good references for like what to use for accelerometry, here is a link to one of their papers that I'm going to drop in the chat. Like, so this is like if you want to start like a rabbit hole of like of decisions to make, and you want to spend some more time sitting down with papers, yeah, maybe just click on a link and follow the first author for that paper, and you'll find a whole a very rich world of information. I think from that. That was pretty much how I got training in this, was following through that. But yeah, but I think then also, yeah, that should hopefully be able to like answer specific questions on that. And then, yeah, I think the paper that I brought up that looked at, so there's a paper that had people wear multiple devices on both arms, and then to see if they went through the same data processing, if you get essentially the same results. And I think either a Fitbit or a Garmin was included on that. So basically, they had them all like kind of like they had like a kind of Frankenstein wrist thing that they put on that people were wearing. And one of those tools was there. So I think like raw accelerometry data, you should be able to like, yeah, get good. There should be resources out there for how to process that, especially with GTIR. You're welcome.
Hi.
Uh,
Um,
yeah, hi.
Hi, I'm Sinyoung. Thank you for all the
Hi.
information. It's really informative. Um, so I'm working with Inzuk, like he asked the question previously. So
Yeah.
I'm coordinating the project of Asian American young adults study in the Midwest, and then we are using Actigraph, the watch here,
Yep.
and then we have 2 more biodata collections, and Dr. Pan helps with us, and it's they are dried blood spot cards and nail clippings, and then
Okay.
we are asking participants to do 3 biodata collections at once. So it takes 2 weeks, and then we are asking them to start wearing watch after 1 week for 1 week continuously. So it takes 2 weeks, and then all the batch, it's gonna be a batch that the collection we do. And now we have 800 participants, and yeah, my question is gonna be more than, yeah, it's gonna be after 5:30, but that's okay.
Oh, yeah. I guess I mean, I am okay with that, but is anyone who's pressing? I think everyone else also feel free to email. I'll drop my email in the chat.
Yes.
But then I will also just keep hanging out here until people are done asking questions. So there's no rush on my end.
Yeah, thank you so much. So it's very like personal consulting question. So we have 60 sleep watches for now, and then it's gonna be a batch every 1 month, I guess. And we are aiming 500. We are aiming to collect 500 participants' data out of 800 participants. Do we need more sleep watch for this? We are thinking of like 1 hour to collect all the data.
So like yeah, like how many? So you want to have like you're essentially having month-long batches, and then you want to get up to how many? You said you had like 50 watches, or was it 60?
We have 60.
60. Okay. I think that should. I think you just need to maybe just pilot to make sure you have a chance to download and then charge them up before they go. Yeah, because how many months do you have? I guess how many months do you have to try to do the study?
Should we do you think it's 5-year project, and then like for this time survey, it's a longitudinal survey, and this time we are thinking of 1 year to collect all the data.
I mean, I think that sounds doable. So I guess for mine, like, yeah, for like one of my projects, like I think we were able to do like with 1 week, only 1 week turn like collection periods. I think that was like 50 people in like a month and a half or 2 months. So like you can do it. It is a lot of work, but if you have a team and like you all, yeah, I think that all of you will be huge experts at both downloading data and charging the watches afterwards. But I think, yeah, and as long as if you have people's like vital information beforehand, like their height and weight and things, like if you want to plug that in, I think height's probably most important because that might change. I guess act to life. I don't know if GGIR really cares that much about height, but yeah, I think that would be enough. But yeah, because just a quick turnaround. You don't need them sitting on the shelves collecting dust. Just put them on someone else's wrist.
Yeah, and yeah, so we are about to launch the survey like next week in 2 weeks. So yeah, do you have any tips in collecting and like coordinating the like watches, like asking them to something, asking participants to do something, or any tips or any things you want to mention when in the
just to make like just to make the handoff quicker, or
yeah,
I guess if they're oh sorry.
no, go ahead.
Oh, are they if they're coming in for like a little bit of an appointment? I guess if you had one person that could take it, and then so you have like one maybe research team member that is making sure they drive through things while the other person does the watch thing and gets it turned over really quick. But I guess I'm trying to like I think yeah, I think as long as you have someone there who can do the watch prep beforehand, and you have like the template like ready with Actilife, I think it's yeah. I don't know if I have too many other tips beyond just having like the template saved, so you can just easily apply it to the next watch like after you download it. Oh, sorry, they're mailing the watches back. I was miss or
Yeah, I haven't mentioned. Yeah, we're sending watches through FedEx.
gotcha. That makes sense. I guess it was like yeah, but I think I've heard I think on one hand, like if people don't get like aren't able to receive like their data or compensation back until you get the watches, like that's one thing. I think also like so I pre-printed my return labels. You've probably already done this, and then like had them in the shipment in the box back, and then also the box itself was reusable, so they could reuse the same box that they got to send back. It might also be a thing to chat with like FedEx if that's who you're using to like have good I think tracking info. So I was using kind of like when I was at UW for my project when I was shipping the boxes, I was using kind of a discount UPS label each time, so I didn't really get great tracking details. I think the tracking will at least help you make sure that they're not getting lost. And then if someone says like, I think it says that they shipped it back, you'll actually be able to know. And then if you could maybe put like the name of you, if your university has like tags for like pieces of equipment that belongs to the university, I think just having something like that on the watch too has helped. Like I've had some collaborators that if you just have that on there, then they think someone is more likely to be like, oh, if I lose this or don't return it, it's actually a really bad idea. It's a really bad thing for me to do. So it's like just because I think it offers some like makes it feel like more official to have it, and then they're like, I really need to get this back, otherwise, like that happens. I've had a collaborator threaten people with legal action. I think that might be a little extreme, and I don't think that they were going to follow through with it. But I think like an easy one might be just adding a sticker. Another one, like I think beyond that, even maybe if there's if you can look up like the nearest Dropbox to them, so you can actually tell them, hey, here's a Dropbox. You can just put it there. Might be helpful. I don't know. I don't know if these all might be things that you've already thought through and worked through. So I apologize if I'm just explaining back to you your like things you've already thought of and gone past.
Well, stickers and letting them know the nearest Dropbox is really, yeah, helpful. Thank you so much for.
I have another
Feel
thing
free.
to add because we've done a fair
Yeah.
amount of shipping. And it was actually recommended to me, I forget by who, but to put your return address as a sticker inside the box. So if for some reason the outside sticker of the box gets damaged, that's available on the inside so that if someone opens the package, they can see where it needs to go to. The other thing that we've had problems with is when people have moved or they have mail forwarding, and sometimes the devices get forwarded to a different address of theirs. We just had one that went down to California instead of Seattle. So just making sure that you have people's up-to-date addresses as well. And then I think just as with everything, just really clear instructions. Like Rob was saying, like where do they have to physically go and drop it off? Or are you scheduling a FedEx pickup for them? Just make sure that that's clear.
That's really helpful, Kendra. Thank you.
Thank you so much.
Like that mail forwarding is really like, yeah, and especially because I was working with when I was shipping with college students, and sometimes they were at their dorms and then sometimes they weren't. And then it's really easy to lose the shuffle of those things if you are not like ahead of it. That's a really good idea. Thinking about like, yeah, mail forwarding and why, yeah, and all those things. Okay. Any other questions or tips that people had? I know we're at the time, but
I have a quick I guess analysis related question. So in a case where let's say we have multiple batches, right? So the first batch we collect 50 people, and let's say that takes for a year since we are collecting, let's say 500 people in total. During the analysis phase, have you seen issues with those who took early? I mean, when we collect early data collection versus those later, do you control, like do you control for that factors, like the time differences? Like how does the literature typically address, or even do they care?
I think it might, if things happened around, like I guess for like one of mine, like it was happening throughout the pandemic, so people like it's like social distancing was lifted during it, and so like because I was interested in some relationships with biology, I had to kind of like I think I mostly used it as a covariate, like the timing of the pandemic, just in case. And like and I think just looked at some descriptive stats and not like showing to see if there was anything I really noticed that changed in the relationship. But in terms of like the literature, I think people usually just, I guess, usually just kind of treat it all like as one batch. I think, unless you're looking for like
okay.
pre-post effects. I think if you're using GGIR or even ActLife, actually making sure that they're all actually this, yeah, like because if software updates happen in the middle of it, like ActLife, you might not know exactly what happens with it. So if there is a way though of saving the raw data, so you could reprocess it. So make sure that everything is processed with the same version, maybe of whatever your software program is. That should be fine, I think. Other than maybe if something happens to like if one or two like if the accelerometers, I don't know. I guess the short answer to like what your actual question was, I haven't really seen much, I think, in the literature to
So they just ignore the time differences, like the data collection differences.
Yeah, like unless you think like unless there was something that changed in like the instructions participants are given. maybe not.
I
Yeah,
mean,
because like
because like,
right now,
yeah,
oh, if I'm understanding correctly, it was like you're asking like if you give people like the same protocol to like last year, and then you had a different set of people get the same protocol this year,
right.
if you should control for the year that they.
Because like it worse, like in the extreme case, let's say we had COVID in the middle of data collection, right? So those who we collected data earlier on didn't experience COVID while there. And then, I mean, in that case, we might see whether the differences, or we could include indicator as a time period, time event, something like that. But in general, because as we were collecting throughout the year, I was a little bit of concern those who like participate earlier on, like for example, in January versus those who participate in February, I mean in December, would experience a lot of different things during that process. So I was just wondering how researchers typically address that issue, or they just um, ignore.
Yeah. Okay. I think I misunderstood part of your question, I think before, because like I wasn't, seasons, seasonality, especially like, throughout the US, like we're going to have some places where
Right.
when it's snowy, your behavior is going to be a lot
Oh yeah,
different than
that's
in the
right.
summer
That
or
too.
spring. Okay. That's what, okay.
Right.
Like, yeah, I don't know if someone has had a good response to that other than maybe like, I think you would just have to like look into it to see like if everyone in winter has, say, like lower activity, then like you could try to figure out some way to account for that. I think the COVID question is always like I've even seen people trying to grapple with that with like inflammatory data and like because they've had some like labs have had higher CRP levels since the pandemic in most of their people and they're not sure why. But I don't know if I've run across a good something that strikes me as like the obvious solution to this.
Okay.
Yeah.
But at least it has a date indicator, so maybe date or time, some sort of time indicator.
Yeah, like you could try it. Like I guess maybe. I mean, and once again, you've probably already like if you've already thought about this. I'm sorry if I'm just repeating things you've already worked through. But like I guess like if you like had like winter or like you could even have like location and time maybe too might be a thing. Like if someone is in like Nashville or Florida, like maybe they'll have fewer changes seasonally, or maybe LA. Like in Southern California, there might be less changes throughout the year than say like Minnesota or Chicago, where like you can get snowed in. But that's a really good question. Like, I don't know. I don't think I've run across someone like actually working through that, like working through either of those bigger questions, like the time, like over years or throughout the year. At least for like trying to explicitly control for it or like appropriately analyze it, their data.
Thanks. Yeah.
Yeah.
It might also be worth looking at sleep data for actigraphy stuff because they factor in a lot of seasonality and also like daylight savings time. So they're thinking a lot about both location for latitude and longitude for daylight savings and also like the season of the year and how that impacts people. So it might be worth looking at that.
Right.
I have a question as well, if that's okay, Rob.
Well, yeah, I was just going to add on it. I just wanted to say that was a great idea that you just brought up. And then also, that reminds me that there's also the light sensor on the actigraphs too, which could be helpful for the time of year thing. But yeah, thank you for that.
The question that I had is sort of related. So I'm working with actigraphs, but in a preschool population, looking at their sleep. And preschool kids move a lot differently than adults. And a lot of these devices have been validated and also used primarily in adult populations. And so the way that their movement is captured mimics an adult, but it has to be translated to pediatric sleep. And I imagine that is true for other populations as well, both age and also other demographics. But do you have any experience sort of translating, like once you have the raw actigraphy data into other metrics, like a pediatric metric instead?
I haven't had to do that stuff yet, no, but I think but I have seen I've seen some analyses and reviews on like basically just trying to build up recommendations for it, but I don't think that they're addressing the question that you brought up. So I don't have anything off the top of my head for that. But that is that's a whole thing that I didn't even get into in this. That is a huge a huge question. But yeah, I'm sorry, I don't have anything off the top of my head.
Okay. No worries. Thank you.
If there aren't any other questions, thanks again, Rob, and we'll send out an email with the paper you shared and your contact information along with the recording of this workshop. So thanks again, everybody.
Thank you.